During embryonic development, budding is controlled through the coordinated development of anterior-posterior and posterior-dorsal axes. Signs of budding first appear at somite stage 16. When limb bud fields develop, they are initially composed of cells within the lateral plate mesoderm, located in specific positions in the flank of the embryo. These cells engage in active cell division, creating a region of rapidly proliferating mesenchymal cells, encased in an ectodermal jacket. Here I show the development of the forelimb and associated somites 15 to 20. Somites are tissues involved in the allocation of the limb fields and limb induction. Prior to stage 16, around the 12 somite stage, the FGF10 gene is widely expressed in the segmental plate and in the lateral plate mesoderm. FGF10 is gradually restricted to two regions along the lateral plate mesoderm, the prospective forelimb and hindlimb areas. FGF10 expression upregulates Wnt3a in the limb ectoderm, activating FGF8 in the ectoderm by intracellular mediators B-catenin and TCF. FGF8 signals back to the lateral plate mesoderm, maintaining FGF10 expression. Budding begins at stage 17, with the pre-AER formed from Wnt3 and FGF8 signaling. Later, at stage 18, the AER becomes defined. The AER, or apical ectodermal ridge, is a thickened ectodermal layer which serves to increase mesodermal proliferation in subjacent cells, labeled the progress zone. In addition to stimulating growth, the AER prevents distal mesodermal cell differentiation. FGF8 secreted from the AER assists to inhibit differentiation, allowing for limb outgrowth. At stage 21, FGF8 induces a zone of proliferation at the posterior margin of the budding mesenchyme. The sonic hedgehog protein is secreted from the zone of proliferation and controls expression of target genes, such as bone morphogenic protein 2 and gremlin. This pathway is mediated by foramen. The gremlin protein production in the distal mesenchyme maintains the apical ectodermal ridge. Interaction between foramen and gremlin to regulate the sonic hedgehog FGF4 feedback loop occurs. Feedback loops between FGF4 expression in the posterior half of the apical ectodermal ridge, in addition to other FGFs, and the anterior bud mesenchyme, help maintain sonic hedgehog expression.